All right, for the next talk, what I want to do now is talk a little bit about therapeutics, and what I'm going to do is sort of follow the initial pattern of the presentation I did, where I was talking about some of the headlines in FSHD, and I had flashed some up, including the Fulcrum trial and the Acceleron study. And so I'm going to talk about those, but as some of you know, I work hard on trial design. It's a lot of what I do. And so I'm going to kind of do a hybrid talk. I'll give you some results from the studies, but I'm also going to take the opportunity to, oh, can you see the screen?
No.
Oh, no, I'm sorry.
Just see you.
All right. Yep. There we go.
There you go.
All right, fantastic. And so what I want to do is I want to start with the Acceleron study. And some of you may have seen this. Some of you may have participated in it. They announced their top-line results. This was a big deal for FSHD. I mean, this is the first phase 2 large industry trial we had in a very long time. And so what I'm going to center on here is I'm going to center a little bit on their choice of outcome measures, their inclusion criteria, and the interpretation of their study. I do have to say before we start, I do consult for companies, but everything that I'm presenting here is public. And if slides are coming from the companies, they'll be marked so you can know that this is something that they've released. I sort of call this portion of the talk, when is a positive study negative? Because as some of you may know, the actual Acceleron clinical trial was a positive study in that it did meet its primary endpoint, which was to look at changes in muscle volume with injection of their drug. So ACE-83 was the drug. It's an anti-myostatin drug. Myostatin is something that halts muscle regeneration and growth in our bodies. It's a natural homeostatic mechanism for us because our muscles are always being damaged and having to regrow. And so there needs to be checks and balances. And so the thought is, in a muscle disease, blocking that pathway could let the muscle grow, and this might lead to increases in strength. The company had decided to use muscle volume as their primary outcome measure for their phase 2 study. And the reason why is well, if you're thinking that your drug is going to make muscles grow, then seeing the muscles change in volume would make sense. They didn't have a lot of evidence for the use of MRI volume in FSHD because there really just hadn't been a lot of studies, but they did have some data from studies of drugs and other diseases like Pompe disease. And they had done an early phase study in people who were healthy. And in FSHD, there was a literature of DEXA, and some of you may know DEXA is a way to look at body composition. One thing that it looks at is lean muscle mass. There's a very high correlation between DEXA measurements of lean muscle mass and measuring it using an MRI and muscle volume. And there's this old sort of study here where they compared strength in the thigh to the lean muscle mass in the thigh. This was from DEXA measurements, and you can see there's this correlation. So the thought was, if we could increase the thigh strength, you know, if we can increase the thigh lean muscle mass, the strength itself may also go up. So there was this relationship between the two that suggested they might be connected. The study itself was in 2 parts. The first part was called a multiple ascending dose study. This is just a study to figure out what the proper dose would be and whether it's safe to give the drug. It was directly injected by needle into your tibialis anterior. That's the muscle that helps you lift your toe, or your biceps, which is the muscle that helps you bend your arms. The second part of the study is a traditional placebo-controlled study. This means that people either were going to be on placebo or drug. They used the dose that they determined from that first part of the study. which was the largest one, the 240 milligrams. They did it bilateral, so you either entered and got injected in your biceps or your TA, but you wouldn't have known whether you were getting the drug or something else. When we look at the inclusion criteria, they actually used a measure of strength to help select people. And so they made an assumption here that the best muscle to measure would be one that was affected but could still move. So they had this degree of strength. Either you had to have some weakness, for example, in the ability to dorsiflex your ankle but still be able to do it, or you had to have some weakness in bending your arm but still be able to do the task. If you look at their primary outcome measure, this study used a biomarker, so MRI muscle volume as the readout, but they did include a broad array of functional or strength outcomes that they also looked at. I would say this is an appropriately designed phase 2 study, looking at a biomarker because they're nervous that in the short time frame that these other measures may not change, but including them so we could see if there's any signs of them being affected. This was the result from the first part of their study. And the way you can look at this chart is you can just, it's the change as the percent change from baseline in the muscle volume. The gray bars are sides of the body that in that first part didn't get injected, and the colored bars are just those increasing doses. And so in the first part of the study, they saw a dose-related increase in the muscle volume, and it was actually pretty big. So people, they were seeing maybe 15% increases in the muscle volume on the first part of the study. In part 2, and this was 58 individuals who entered this, half were in the biceps, half were in the TA, they saw the same increase in muscle volume. And so when you compared treated people to people who had the placebo, there was about a 9 to 15% increase in the muscle volume that you didn't see in the other ones. This was significant in the population. And so the drug reached its target and it made the muscles larger. They also saw a commensurate decrease in the fat fraction as measured on the MRI in one of the muscles. And this is an indirect measure of the state of the muscle. As we've talked about today several times, that as muscles become affected, you do see fibrosis and fatty changes in the muscle, and this is a way to try to measure that on MRI. So in terms of MRI, this was a positive study. But the functional testing, they didn't see a strong signal that the drug was working. And so the company actually halted their development program, and they said, well, we don't see a path forward. And what caused this is just showing you one of those functional outcomes. This is the 6-minute walk test. Some of you may have had the pleasure of doing this. It's aptly named. It's just how far you can walk in 6 minutes. And this is just showing you the change from the first time that people were seen. And then over time, the red is the treated group, the gold is the untreated group. But I think the most dramatic thing you're probably noticing here is that actually both groups got a lot better compared to the first time they were measured. And so the company was like, they said, well, we didn't anticipate this. This is an interesting phenomenon. It has been described in FSHD before, and it's also been described in many other diseases that are sort of chronic and progressive over time, which is there's an effect that happens to people when they enter a clinical trial where their performance can improve. And what I'm showing down below here is some data that we had published many years ago, where we had looked at those prior clinical trials that happened in the late 90s and around 2000, and we compared them to that prior natural history study. Instead of looking at a functional thing like the 6-minute walk test, we were looking in changes in average strength over the whole body using 2 different techniques. The blue line is what we see if we don't treat people, that slowly over time, there's on average a decrease in strength. The color bars include both treated and people on placebo. And I think what you can see is that with these two different measurements of strength, there's an increase in strength that occurs when people enter the clinical trial, whether they're on placebo or not. And here you can see that it starts to wear off at about 12 months. And so there's a number of important lessons that we can take from the Acceleron study, and I think it's important for the field as we move forward. One lesson, and I always say this when I'm consulted for a company, is that you need to make sure that your study is powered to answer the question you actually want to ask. And in this case, they had the right number of people to see a change in muscle volume, and so they got that answer, but the answer the company really wanted was whether it was going to improve function. But they didn't really have the right number of people in the study to answer that question. They didn't have enough people. They would have had to double or triple the number of people in the study to really be able to see that over 6 months. And so one thing you need to do is make sure you really are asking the question you want to answer for. And while a biomarker can be essential for these early phase studies, because like I said, it's often hard to look and see a change in function because it can be slow or require a large number of people, we have to ask if we really know what a change in our biomarkers is going to predict over time. And there's a good example here because they saw a change in muscle volume, and they've assumed because they didn't see the change in function that it wouldn't occur. But if you can imagine if we had a natural history study where we could have shown that a change of 10% in someone's muscle volume had some predictive value on whether they were going to require, say, a wheelchair for ambulation in 10 years or not, well, you might have taken the results of this study and said, well, we don't care that the function didn't change in 6 months. We might still want to take that drug because we would understand the predictive value of the biomarker. And so as we're thinking about our future clinical trials and getting ready, it's important that we understand the tools that we're going to use and understand how large a change in these tools, what it may mean clinically over time. And these are questions for us, but questions that we can definitely answer. I want to switch gears a little bit now, and I want to talk about the Fulcrum study. The Fulcrum study is a very interesting study, and if you've been in it, you know it was somewhat complicated. The inclusion criteria were a little confusing, and so I want to use this as an example of how you start to think about designing inclusion criteria for a clinical trial and how it really depends on the question that you're asking and the drug that you're wanting to test. So when we're thinking about trial design, there's sort of two things. There's outcome measures and there's the design of the study. And people often ask me questions like, what's the single best outcome measure, or what's the single best trial design? But the truth is, this isn't like the Lord of the Rings. There's not one to rule them all. The truth is, it's going to depend. The outcome measure depends on the mechanism of your drug and what you're hoping your study is going to show. And your trial design depends on where you are in the development of that drug. For example, when we're just starting to test a drug in humans, we're actually most interested just knowing if we know the dose and whether it's safe or not to give the drug. As we go further along, we start to ask more complicated questions. Is the drug reaching its target? Is it having an effect? And then finally, as we get closer to marketing, we really just want to know, is the drug helping people? And will it help people who have, a broader population who have the disease? So Fulcrum's study was a phase 2 study. The drug was losmapimod. They performed a randomized placebo-controlled study. Originally, it was planned for six months. And their primary outcome was going to be a change in muscle DUX4-related biomarkers. So another study where they've decided they're going to look at a biomarker as their primary outcome. So what is losmapimod? It's a P38 or a MAP kinase inhibitor. It's a class of enzyme that has really broad effects. The hypothesis was that it was going to have a direct effect on DUX4 activation. And the question becomes, how do we know this? And I think one lesson you're getting out of today is that models for the disease are really important if we're wanting to have therapeutic development, because we need to be able to know, does a drug have a likelihood of having a positive effect if we're going to give it to people? And the way they got their information for this study was using patient-derived cell assays. And this is just an illustration of how we go about doing that. We often start with people who have the condition, so who have FSHD. In this case, we would take a piece of your muscle. We would do chemical things to the muscle to move it earlier in the process of differentiation so we could grow it, and we could grow it repeatedly. And we might change it a little so we could see the output we really wanted to see. And for FSHD, that output is the expression of DUX4, or if not DUX4 directly, then those genes that DUX4 is acting on. We can then take those cells, put them in a petri dish at that, 100 or 1,000 at a time. We can use computer techniques to expose those different little wells to different kinds of drugs that are in established drug libraries that you can purchase. They have usually several thousand drugs at a time. And then you can use automated processes to look at the readout as long as you've got some kind of reporter, DUX4 or one of those gene targets, and look for drugs that might be having an effect. And so this is what they did to identify losmapimod. In this case, you can see that the readout they were looking for was DUX4, but also a couple genes that are directly, that DUX4 has a direct action on. And you can see that if untreated, their levels are very high. And so what they were doing here, they were showing that if they expose these same cells to their drug, losmapimod, at different concentrations, suddenly you could knock down the expression of DUX4, but also those other targets of DUX4, suggesting your drug was reaching what you wanted it to reach. And you can see they use studies like this to help them pick the dose that they were going to study in human beings. Now, you're also interested, though, if a drug's going to have an off-target effect. And so the value of these preclinical models is you can also look for markers that tell you off-target things you may not want affected, in this case, muscle growth or regeneration. And they're showing that their drug doesn't really have an obvious effect on those other measures of muscle regeneration. So when we think of their clinical trial, they wanted to show losmapimod decreases expression of DUX4. They used a design that was a 6-week randomized and placebo-controlled study. Their primary outcome was the DUX4 targets. And so how are they going to get this? Well, they're doing it by muscle biopsies. They take one before you start getting treated, and they take one after treatment with losmapimod, and they compare the level of these DUX4 targets. But one question that may come up is, how do they know that a muscle is going to show these DUX4 targets? And this is where we get to the importance of inclusion criteria. And some people ask me, because I'm running a large study where I say one of the goals is to help refine inclusion criteria, but the reason you need to do it is because you need to be able to help companies when they're thinking about how to design their studies. And so what did they do? Well, they chose adults. They wanted people to have FSHD, so they chose a genetically confirmed person. But they really wanted to be able to pick muscles that were going to show this DUX4 activity, and they used two things to try to do that. They used a clinical severity score that's commonly used in the FSHD field, and they used the MRI itself to help them pick the muscles that they were going to include. So what are these things? Well, this is the clinical severity score. It was designed by an Italian group. You can see that cartoon I had shown you earlier that just shows us the typical pattern of FSHD. The severity scale literally just follows that pattern. It starts with facial weakness on the less severe end, then the shoulders are affected, then later the distal leg, and then finally the more proximal leg. But the advantage of a scale like this is you can interpret a range on the scale to help select individuals who are likely to have muscles in the legs that are affected. And if we look at the inclusion criteria they chose, which I've highlighted here, how many people would that be in a randomly selected population? This would be about 65% of people who we would see who have FSHD. So how did they use MRI as an inclusion criteria? There's a couple ways we can actually use MRI, and they are doing both in this study. One way is an outcome measure itself. We could use MRI to give us information of the change in your muscles over time. This is just showing you a cross-section of the leg in someone who has FSHD. If you look at it, you can actually see what we call that fibro or fibrosis or fatty replacement, because the muscles go from normally a dark appearance to looking almost bright white. You can use a computer that can actually calculate this as a percent, and we can compare it to things like that severity score we talked about, or strength or other measures, and you can see there's a nice correlation. The higher the fat, the more severe the disease, the weaker someone gets. And so that would be one way we could use MRI. But another way that you can use MRI is to select muscles where we think there's activity related to FSHD actively going on. This is a different kind of sequence on MRI. We call it STIR. It looks at water in the muscle, and water tends to come into the muscle when there's signs of inflammation. And I think you saw in Peter Jones' talk that one of the hallmarks of FSHD, when it's actively presenting DUX4, is that you get some inflammation. And here you see an individual who has one of those STIR positive muscles, and it turns out that if you biopsy these muscles, you get a higher chance of seeing DUX4 or those DUX4 targets when you look in the biopsy. And in fact, if we just randomly picked a muscle to biopsy, only about half are going to show us those DUX4 markers. But if we use this simple MRI signal to help us select, then we start to get up around 95% of the time we're going to see these DUX4 targets. So how do we put their inclusion criteria together? Well, they have adults who have genetically confirmed FSHD, who had moderate to severe shoulder weakness and weakness in some lower extremity muscle. So that's that CSS between 2 and 4, which I told you was about 65% of people, who had a muscle showing a possible signal of molecular activity on MRI, which in this case is about half the people who would meet that clinical severity criteria in the first one. And so overall, what we end up with is about 30% of people who would have screened for this study, if we knew nothing about them, would have had a muscle that was positive for biopsy. And indeed, if we look at their inclusion criteria, because of the inclusion of that severity scale, they got a little higher, but it was about just over 50% of people qualified for the study. And this is just some data from earlier study they did showing these DUX4 targets in the muscle. Over here on the left, you can see these were healthy volunteers, and you can see that they don't show those DUX4 or DUX4 targets in the muscle. Here you can see that the individuals that they chose using those simple MRI criteria are showing evidence for that disease activity, although I think you can already see one issue that we're going to have here, which is that there's a lot of variability in how much of that activity they're seeing. And the other thing to notice is that even with those criteria, there's still a couple individuals with FSHD who weren't showing those markers. So where is the Fulcrum trial at today? Well, they completed recruitment. Unfortunately, we had a pandemic. The pandemic had all sorts of effects on this study, and I'd say it's a real tribute to Fulcrum that they stayed with this study and they've continued it despite all the difficulty of trying to do it in the pandemic. But one thing that they actually had to do was to change the timing of their muscle biopsies from that 24 weeks to 48 weeks. And so there's people still in the study. We're still trying to learn about it. They did do a public announcement, and many of you who are in the study probably heard this, that their initial analysis of the DUX4 biomarkers didn't show an obvious effect of treatment except on the highest DUX4 target expressors. And this is a publicly released table from that analysis. The thing I mainly want to point out here is this is less than half of the individuals. So I always really caution people to look at trial results early. I don't think it's a good idea because of this, because you may not know how to interpret the answer. But you can see here, if we look at the change in these DUX4 targets, that overall there was a very minimal change in the total groups, and they didn't really separate out. When they look on the extremes, though, the people who had a very high level of those DUX4 targets initially, now you can see that there does appear to be an effect of their drug. We'll learn more about this as the study goes on, and what I would say is that there's a lot of other measurements in the study we're going to be very interested in. So no matter what, we're going to learn a lot from this study. There's still a chance that we may see other benefits of the treatment in the study once they reach the end. And so I do think this is an important study for the field. We're going to learn a lot to help us design those future disease-targeted therapies. Now, I know I'm starting to get a little long, and so what I'm going to do is I'm going to just finish up with briefly talking about our own network, because I've talked a lot about trial design, and of course we have this clinical trial network that's run out of KU in Rochester, and it's been growing quite a bit. It started in 2016 with 4 sites. We now have 12 sites in the US. We have 3 sites in Europe, but we're adding 3 more in the next few months, so it's quite a large network of people. We have lots of funders helping us, and Dyne itself, who you've been hearing about now, are also funding this because they want to be able to use this to help them design their future study. When you think about what is it that we're doing on this network to help these companies prepare for therapeutic trials, we actually have more than one study running at the same time. We have a lot of sites. There's a lot of people with FSHD, and so we're able to do this. We have one study called Resolve, and this is really mainly looking at strength and functional measures over time. These would be the kinds of outcomes that a company would need when they're getting closer to wanting to get marketing approval for their drugs. We have another study that's running to look at those MRI and muscle biomarkers that we've been talking about, and it's running at a number of our sites, and we'll be trying to confirm how to use those muscle biomarkers and what changes in the muscle on MRI over time. And we have a new study that we're starting that's a more of a clinic-based study, because we're concerned that there are groups of people that we haven't been able to study well, people who either can't walk or have FSHD type 2 or something that hadn't been included in those earlier studies. Just a brief update on Resolve for those of you who are in it, or those of you who are wondering. The Resolve participants were limited to people with FSHD type 1. They had to be able to still walk independently for 30 feet, but they could use canes or braces to do this. This was designed to match what we thought was the most likely criteria for the upcoming clinical trials. But as I mentioned, there were other people who weren't included, but we're trying to get the data on these people now in a different study. When you look at the things that we're measuring in Resolve, and this is just showing you what we do at each visit and how many visits we have, you can see that we have a number of biomarkers. These are biological fluids where we can look for biomarkers. We have patient-reported surveys or questionnaires. We have strength and functional testing, and we also have some biomarkers like DEXA, our electrical impedance myography included. When we think about what we measure, one of the important measures that we included in this study was an FSHD-specific composite measure. The way we came about doing this is we actually started with surveys of people who have FSHD. We asked them what was important to them and how impactful it was on their life. We then took those categories and had our trained physical therapists look in the literature and find ways to measure those different categories in the body. And so it's actually not one thing, it's actually 17 separate types of tests that we do on someone that we can then combine together. this is just some data from the study to date. And the reason I wanted to show this wasn't to spend a lot of time going through charts or graphs, but I want to talk about why a study like this is so powerful. And it has to do with who's included and how deep you can go when you start to think about the analysis. And I just want to point out a few things. That a study like this has a lot of people, so 200 so far. We're ultimately going to have close to 300 people in the study. When you look at our age range, it's the full adult age range, the full range of when people started to become symptomatic. When we look at the genetics, what I would say is this is the full range of genetics that we see in the population. These are severity scores, and what I would point out is that we're covering the full range of severity that we see on those scores. When we look at our strength measure, which is this MMT, we're seeing a very full range of strength as well as a full range of function. So one thing that's important is we want a broad enough range of people so we can ask questions about those inclusion criteria. Doing this also gives you some real power to think deeply about your outcome measures. This table here is sort of interesting. If you look on the left, these are individual muscles, and this is just going, the top is your hips, the bottom is your ankles. So we're going from the muscles in your hips all the way out to your ankles, and we can look at all these different types of tests we do, the 6-minute walk test, the 10-meter walk run, a time to ascend stairs, and we can say, well, what muscles are important to guide these outcomes? The yellow and the green color are highly correlated, and I think you can see one of the outcome measures is really pulling itself out. It has most of the muscles affected in the hips to the ankles having correlations for people with FSHD, and in fact, you can see two muscles, the hip extensors and the knee flexors, where it's quite strong. These are muscles that are related to burst performance, acceleration. And when we look at what happened to people over time now, this is the change in that measure, the 10-meter walk run at 3 months, 12 months, or 18 months. You can see we're seeing progression of FSHD over time. And these are significant changes for the individual, albeit small. And so this is how you start to build the argument for a particular test being important to use in something like a clinical trial. So what are we doing? We're expanding our study into Europe. We're still completing the RESOLVE study. If you're in it, your help is very important. And it's all these follow-up visits that we do that give this study such power. We have the MRI and muscle biomarker study that's going on. We have a clinic-based study we call MOVE. And in the future, we are really interested in supporting clinical trials of repurposed drugs. And I think you heard a little about this earlier from Peter, where everyone's excited about the gene-based therapies. But I think an area where we have the possibility of identifying treatments that have a shorter pathway to being available to people is to look for drugs that are already out there that may have a very important effect for FSHD. And so we as a network are looking for those and hoping to test those to sort of bridge that gap between where we're at now and where we'll be when we have those gene-based therapies. All right, thank you very much, and I'm happy to take any questions.
I have a quick question for you.
Sure.
You mentioned the 2 studies that you're currently recruiting. Do you have information on recruitment criteria somewhere on a website or something?
Yes, we do, Kylie, or I can let anyone answer that. Yeah.
Yeah, like how to get into a study?
What criteria are involved in inclusion, exclusion?
Oh,
criteria?
yeah. So our studies are posted on clinicaltrials.gov, but you can also, email Michaela and I and ask for information. We also have an FSHD CTRN website that posts, we post our studies on there as well. And I believe the FSHD Society also has our study information posted on their website.
But we're always happy for you to reach right out. Oh, go ahead.
No, go finish your statement. I can retain my question.
Oh, well, what I was saying, we're always, you can just reach out to us directly. We're always happy to answer questions.
Okay. So is there a discussion about the possibility of a tolerance to the losmapimod over time, that the body builds a tolerance and that the disease starts to take over again over time? Is that in discussion or a possibility?
Well, I think what you've done is you've asked a very good question. And so I think what you're saying is, while losmapimod may have that effect on DUX4, it's certainly possible the body has its own mechanism to minimize its effect over time. And
Better stated.
Yeah, well, no, I mean, I think it's a fantastic question. You know, and this is where animal models would have been very useful, but I think you may remember that when Peter Jones was discussing the mouse model, he said, well, it's good for certain types of therapeutics, but losmapimod is going to be in a category we can't really test in that mouse model because the mouse doesn't have the human regulatory pathways for DUX4. And so we don't know the answer to it. And so the answer is, yes, it's possible that would be true, but we just don't know right now. I think right now we're asking a more fundamental question, which is, can we see any benefit for giving people the drug? And that's what we're really hoping to see from this current study. But yeah, an equally important question.
Dr. Statland, I have a question. Yeah. You know, with the pandemic, there have been, you know, research going on every corner, every place in the world for this pandemic. And I realize it's a whole different type of cure that they're looking for, but we don't really know, I don't think, how their research is being handled, what they're looking at with that research. Do you think that there's any way that the studies that have been going on trying to find a cure for the virus may uncover through some way a possible drug that may be beneficial for solving or treating the FSHD or other gene issue? And if so, are there links to the different researches that are going on that might be able to provide that insight?
So an interesting question. So I think the question, we could take it even broader and just say, how much crosstalk are there from our different diseases in what people are exploring? There are ways there are ways to do that. Now, unfortunately, some molecules are truly proprietary, right? Someone really discovered it. It's not publicly available, and so we couldn't really test them across different diseases. Some of them are available in libraries. So when I mentioned those large libraries you can test, they actually contain many thousands of drugs that have been developed for other diseases that currently aren't on the market. And that's one way to ask that question. We can take it into our FSHD model, run 1,000 of those different compounds and say, do any of them look like they're going to work for FSHD as well? And so, yes, there is a way to do that. It also goes the other way. I don't know if you know this, but losmapimod, which we're testing for FSHD, they're also testing it for COVID. So things can go both ways. It turns out that this drug was initially an anti-inflammatory drug and has an effect on that pathway. And so there was a rationale for why it might be important for the bad side effect, you know, the bad outcomes people were having with COVID. And so it does go both ways, but, you know, so the answer is yes. I don't know that, you know, certainly not on probably the vaccine side, which is a different concept, but in the disease altering therapies, it's possible some might cross over.
Well, that was actually what generated my question is somewhere in the last 7-8 months since the beginning of this, I did see that there was that crossover where those therapies were drugs were being used, you know, from one to the other. So that was kind of the beginning of my thought process. So thank you.
Yeah. Well, remember, one of the effective therapies we have for the moderate to severe COVID to prevent complications is steroids, dexamethasone. this is widely used already for multiple disorders of inflammatory disorders. So the truth is, this happens all the time where we take one drug that's used for one thing and we try it in something else. And we're certainly interested in the dystrophies in doing the same thing.
Thank you.
Yeah.